Hi, I'm Dave Moran. I'm the co-author of the Visual Histology DVD series. This film clip is to give you a very brief introduction to what the DVD series is and what one of the individual DVDs is like. First of all, there are a total of 26 DVD programs in our Visual Histology series, and each one's dedicated to covering a specific area of histology. The format of each DVD is consistent throughout the series. We give you about a 5-minute introduction to the material, and then we describe images that you see with the light and electron microscope in some detail for about 20 minutes. And then we give you a 5-minute practical exam at the end so you can test your own knowledge and see what you've learned. Taken together, the 26 programs in the DVD series provide you with a complete basic course in histology. What we're going to do now is give you a 3-minute preview of program number 7, which is on muscle. And remember, this is a highly compressed preview because the whole program normally takes 30 minutes, and we'll just show you about 3 minutes of it.
This is a much higher magnification view of a longitudinal section through a skeletal muscle. Remember, skeletal muscle is also called striated muscle, and in this image it's easy to understand why. Skeletal muscle is characterized by these longitudinal striations that represent banding patterns created by the filaments of which skeletal muscle is made. Now, in this micrograph right here, the greenish material up here is connective tissue of a tendon. The reddish material down here is a muscle. This is taken from a muscle-tendon junction of a monkey. In this image right here, you can see the banding pattern of skeletal muscle very clearly. You can see that there is a thick, dark line, followed by a clear zone, followed by a thin, dark line, followed by a clear zone, followed by a thick, dark line. Each of these bands has a name. The names are much more easy to understand if you look at an electron micrograph of the same material in which the filaments which generate these banding patterns can be resolved. This is an electron micrograph of a longitudinal section through a skeletal muscle fiber, and only a very small part of the muscle fiber is shown here because, remember, these muscle fibers are huge, and the electron microscope, which operates at high magnification, can only show you a very small part of it. So far, we've looked at two kinds of muscle. We've looked at skeletal muscle and we've looked at cardiac muscle. Now we're going to look at the third kind of muscle, which is called smooth muscle, and as the name suggests, it's radically different from the other two kinds in that it has no cross striations and hence looks smooth under the microscope. Now, above this arrow, you see smooth muscle fibers which are cut in longitudinal section, and below it, smooth muscle fibers that are cut in cross-section. And what you can tell right away is that smooth muscle fibers, when compacted together in bundles in organs, look very different from the individual smooth muscle fibers, such as the one we just saw, which had been teased and put into solution. What we'll do now is proceed on and give you a practical exam, which will consist of 10 questions of images you've seen. Question number 3. In this image, what is this structure the arrow is pointing to right here? What is the structure the arrow is pointing to? If you said it's the nucleus of a skeletal muscle fiber, you're absolutely right. And here's another one over here. The next question would be, what is this?
Well, that concludes our 3-minute preview of the Muscle DVD. Thanks very much for watching it. And if you have any questions, just give us a call or email us. Our contact information is on the website, and we'll be happy to talk with you. Take care and have a good day.